everyone. For the journal club topic, I chose the biomineralization inspired mineralized hydrogel promotes repair and regeneration of dead in bone and heart tissue, which was published in 2023 in the journal NPJ Regenerative Medicine with an impact factor of 6.4. So coming to the unique terminologies that we need to discuss before discussing this paper. So treated dentine matrix, what is it? It is a powdered form of dentine, clean and prepared to help new bone and tissues to grow. Shear thinning property, it is the ability of a material to become more fluid when squeezed or pushed, like a form of toothpaste, making it easy to inject in a environment. And supramolecular self-assembly, it is a natural process where molecules organize themselves into larger structural materials without needing chemical bonds. The material that developed had this supramolecular self-assembly properties. So coming to the novelty, aims and objectives, the researchers wanted to develop a biomineralized inspired hydrogel combining PAA, CMC, ACP and TDM for heart tissue regeneration. It was a self-healing injectable hydrogel scaffold that fit into the irregular shaped bone and tooth defects, enhancing the bone regeneration activity, maintains biological activity of natural dentine matrix while improving the mechanical stability and injectability as a whole. It had both osteogenic and osteodontogenic, that is bone forming and tooth forming effects in vitro and in vivo that they tested, and offers a non-covalent dynamic network scaffold design as a novel route for dental and osteopathic tissue engineering. So coming to the aims of the study, they wanted to design a biocompatible and bioactive hydrogel that mimics the natural heart tissue mineralization process to improve the mechanical performance and the delivery of the injectable hydrogel that they tested, to test the tissue repairability of the hydrogel for bone and dental defects in preclinical models, in vitro and in vivo. So objectives: formulate a hydrogel containing these complexes, characterize the hydrogel structure, mechanical strength, swelling, degradation properties and injectability, evaluation of the characterized hydrogel, how it affects the cell migration, proliferation and differentiation of the dental pulpal stem cells. Test this for bone regeneration in animal models. They use the rat femur, skull defects and dentine formation in dog pulp capping models specifically, and they compared the hydrogel performance with traditional TDM powder and a commercial pulp capping material which is iLute BE+ that was easily available. So coming to the materials and methodology, a special injectable gel was made using natural tooth powder, jelly-like chemical PAA and sugar-based polymer, that is CMC, along with calcium and phosphate salts to help form minerals. Lab tests were done using dental stem cells to check if the cell helped in the cell growth and the mineral formation. Animal experiments were done in rats and dogs and showed that the gel could repair the damaged bone safely and effectively. So the timeline of experimentation was, when they did the cell proliferation, migration and differentiation assay, it was done at 7 and 14 days. In vivo, when they tested in rat models, subcutaneous implantation for biocompatibility was evaluated at 7 and 14 days respectively. Bone regeneration in the femur and the calcaneal defects was assessed at 3 and 6 weeks, and dentine bridge formation was seen at 8 weeks, approximately 2 months from the start of the experiment in dog pulp capping model. So they used one-way ANOVA to have the statistical analysis, and they compared parameters such as cell growth, mineralization, gene protein levels that were produced, gel strength, swelling, degradation, bone dentin healing and angles. So this injectable hydrogel combination showed better healing, stronger structure and safer tissue response compared to the controls. So coming to the results, this PAA, CMC and TDM hydrogel demonstrated favorable injectability, self-healing capacity and adaptability to irregular defect geometries. Mechanical strength characterization and structural characterization confirmed for its stability, interconnected porosity and resistance to rapid degradation rates. Also, the in vitro assay showed that it supported the viability, migration and the osteodontogenic as well as osteogenic differentiation of the dental pulpal stem cells. It upregulated the expression of the various markers such as DSPP, DMP1, OPL and RUNX2. These are the odontogenic and the osteogenic markers involved both at the gene level and the protein level. In the rat femoral and the calcareal defect models that they developed, this combination promoted substantial new bone formation with improved bone turnover and trabecular number compared to the controls. In the beagle dog pulp capping model, the hydrogel facilitated the dentine bridge formation at 8 weeks and it was compared to the clinically approved material IUBP plus, and it had various advantage. Histological and immunohistochemical studies indicated a mild transient immune response, confirming the material's biocompatibility and biosafety in vivo. So coming to the strength of the study, they had developed an innovative material design which was injectable, which had self-healing property, modality and bioactivity. It was multifunctional in nature because it promoted both the osteogenic differentiation and the odontogenic differentiation, making it suitable for dual applications in orthopedics and in dental applications. They had done a comprehensive evaluation starting from physiochemical characterization, in vitro cell assays to in vivo animal model studies were done. It had demonstrated clinical relevance in pulp capping and critical bone defect repair both in the dog and the rat models, highlighting its potential for real-world application and therapeutic use. And it was biocompatible and safe because it exhibited low immunogenicity and stable degradation over time. So coming to the limitations, so it did not include long-term in vivo evaluations after 2 months to assess the durability and integration over extended periods. Mechanistic pathways behind the stem cell degeneration and the mineralization was not fully explored at the molecular level, so we need to have molecular level studies in future. In the large scale human clinical trial, the evaluation of the hydrogel performance remains untested, so we have to do human clinical trial experiments. Batch to batch consistency of this component may vary depending reproducibility. So no comparison with other advanced biomaterial beyond this IUBP plus was provided. So comparisons can be done in further studies. So coming to the study guidelines, it is a preclinical experimental study involving both in vitro experiments for the stem cell assays and in vivo animal models, as I already told, for rodent, rat and beagle dog pulp capping model. So it reports, the study reports that ethical approval was obtained for both the animal use and the human derived cell tissue samples. The study follows the standard preclinical evaluation protocols for control group comparisons, replications, use of the statistical analysis, histological imaging and molecular assessment. But there was no mention of the use of the ARRIVE guidelines for animal research, which are often recommended for animal studies. So coming to the future directions, long-term in vivo studies are needed to assess the hydrogel's durability, biodegradation profile, molecular pathway analysis and gene expressions should be done in a broader level. Clinical trials for the human relevant models are necessary with this combination for both dental and orthopedic application. Standardization of the TDM preparation should be done to ensure batch-to-batch consistency in future. Comparative studies with other bioactive materials, that is bioactive ceramics, commercial scaffolds that are available, should be done to help benchmark the efficacy of this hydrogen, and incorporation of additive biomolecules such as growth factors or the antimicrobial agent would help further enhance the regenerative outcomes in the future studies.